Hello, I'm Chulhee Choi, the CEO of ILIAS Biologics. I'm very happy today to introduce our recent progress on exosome engineering for active targeting and intracellular delivery of therapeutic protein in this wonderful meeting on behalf of all our ILIAS team. So my talk will be divided into three parts today. So the first one, I want to focus on our key technology for targeted delivery and active loading of protein into the exosome. And second part, I want to introduce our lead pipeline, ILB202. And if time permits, I want to introduce a little bit about our company. So as we all know, the exosomes are excellent drug delivery system with safe and specific and biocompatible profile. And I will introduce the Explorer, the key technology of our company, loading the large protein drugs into the exosome, and ExoTarget for active targeting. And the third component of our key technology for the scalable production of GMP-grade exosomes, I don't think we have enough time today, but probably next time, I can share this data with you. So first, the Explorer is abbreviation of exosome engineering for protein loading via optically reversible protein-protein interaction. As you can guess from this name, we have adapted optogenetics for active loading of therapeutic protein into the exosome. So we have adapted the protein interaction module that can be modulated by light stimulation in plant. So-called cryptochrome 2 is a photoreceptor that can change their conformation by absorption of specific wavelength of light. And in the presence of blue light, in case of cryptochrome 2, they can bind to their counterpart CIB or CIBN. So what we did here is we made a fusion protein with this photoreceptor with our protein of interest, and the CIBN with exosome targeting protein, such as CD9 or other tetraspanin protein. So by expressing these two fusion proteins, this cargo protein fused with cryptochrome 2, they stay in the cytosol, and this counterpart fused with the CD9 present in the exosome producing domain. By introducing the light in the cell culture system, these two proteins, they bind each other, and then with natural biogenesis of exosome, our protein of our interest can be incorporated inside the exosome. And these cells can secrete the exosome loaded with the protein of our interest. The most interesting part of this technology is after isolation, we cut off the light, and then the protein stop interactions, so dissociate with each other and make the protein of our interest into the soluble in the free form inside the lumen of the exosome. So by this method, we can achieve two goals of our original study: the active loading and loading with protein as a free form in the exosome. So by making the exosome with free cargo inside the exosome lumen, we can actually enhance the delivery of this therapeutic protein inside cytosol of the target cells once they are uptaken by simple membrane fusion, or after endocytosis fusion with endocytic membrane, the cargo can penetrate into the deep side of the cytosol. And this idea has been published 5 years ago in Nature Communication, and IP has been granted in many countries, including China, Japan, and USA. So this is a proof of concept study. So using M-cherry as a reporter protein, we have generated the 293 T cells having the M-cherry cryptochrome 2 and CD9 CIBN by stable transfection. And without light stimulation, they produced exosome without M-cherry protein, even though they have the M-cherry in the exosome producing cells. Only after illuminating with optimal doses of light, they can produce the exosome with our reporter protein M-cherry in the exosome. So when you compare it with other conventional method, we can enhance the loading efficiency by 100 times. And moreover, the idea the free form of cargo can penetrate into the cytosol of the target cell has been proved by treating this M-cherry exosome in the HeLa cells. As you can see here, if the M-cherry is attached to the membrane of the exosome, the pattern of M-cherry should be like a dot shape in the target cell. But as you can see here, when the HeLa cells were treated with our M-cherry exosome, M-cherry signal can be observed as a diffuse manner in the target cells, suggesting that our cargo can be diffused into the cytosol as a freeform as expected. And also we made a luciferase exosome, and by comparing the recombinant luciferase protein, we can measure how many proteins we are able to load into the exosome. So about 5 years ago, we are able to load about less than 2 molecules per each exosome, but right now we can load about 50 molecules per each exosome right now. And also we have shown that active enzyme, here the cyclic DNA recombinase, can also be delivered by our method, and they can retain the enzymatic activity. So to measure the Cree enzyme activity, we introduced the Cree reporter construct into different type of cells and then treated them with naive exosome or our Cree exosome. But as you can see here, only the cells treated with Cree exosome show the robust and the Cree reporter activity. So suggesting that our Cree can also introduce into the nucleus of the target cells. And when we make the exosome loaded with BAX, a kind of pro-apoptotic protein that has been known to induce apoptosis by making the pore outside the mitochondrial membrane. They should act on mitochondria. So when we make the exoBAX, when treated into target cells, they can target the mitochondria of target cells and release the cytochrome C from the mitochondria and subsequent apoptosis in the cell, providing the rationale that our method can introduce the cargo protein into the cytosol and they can find their original site of action, like nucleus or mitochondria. As I've shown, we can load a variety of different cargo protein into the exosome, so making our platform technology for different type of pipelines. So first we can deliver the antibody fragment, so those so-called intrabodies can block so-called undruggable targets. And also we can deliver the signaling molecules, endogenous inhibitors, or transcription factors. And also you can deliver organelle-specific protein, as I've shown in case of BAX or Cree, so they can find their original site for their action. And finally, we can deliver the gene editing protein like CRISPR-Cas9. So we think this exosome-based delivery of this gene editing protein can be a new way for the gene therapy in near future. So I want to gear my story to the second part of our technology, the active targeting part. Before discussing the active targeting of our engineered exosome, we have to know the natural tropism of our exosome. So we are making currently the exosome from the 293 human embryonic kidney cells. So these 293 cell-derived exosomes, when we studied the biodistribution with different methods like radioisotope labeling or fluorescence labeling or other different method, we see almost the same pattern of distribution. So more than 80% of exosome go to liver, and another 10 to 15% go to spleen, kidney, or other immune organs. And even though it has been known that the exosome can pass the blood-brain barrier, but we can find that around 0.1% of exosome, when administered systemically, can pass the blood-brain barrier and reach the brain parenchyma. And we don't see much signal in other GI tract or bone or kind of other tissues. As I mentioned, other than just labeling the exosome with radioisotope or fluorescent dye, we can use this exocrete to study the tropism of the exosome. So by introducing this exocrete into crete reporter transgenic mice, we can know, what kind of cell, what kind of specific cells uptake our exosome by checking the crete reporter activity in the target tissues. And this is one of the examples how we can use this exocrete to study the biodistribution. In collaboration with Texas group, we have studied the role of the exosome in the pregnancy by mating the wild-type female mouse with reporter homozygote transgenic male. So these pregnant mice have a reporter transgenic fetus in this wild type. So only the fetus is a reporter transgenic mice. So this reporter is TD tomato, and once crete is introduced, this crete TD tomato red signal can be switched to the green fluorescence signal. It's a very high signal to noise compared to previous crete reporter system. But as you can see here, the TD tomato signal can be dramatically switched to GFP signal once the crete exosome is administered systemically in the maternal side at day 15. This clearly suggests that the exosomes can pass the placental barrier, at least at the day 15 of pregnancy. The day 15 of the mice correspond to almost 25, 27 weeks of pregnancy in human. So we didn't know at the time whether the exosome can pass the placental barrier at the time of the pregnancy. But we have very strong scientific evidence that exosome can pass the placental barrier. This data suggesting that our exosome can be a good therapeutic delivery tool for fetal compartment disease. So I'll come back to this idea later. So by displaying the tissue targeting peptide on the surface of the exosome, we can achieve the active targeting. So this idea is so-called exotarget. So as one of proof concept study, I will show you our recent study result. So by displaying the BBB specific peptide on the surface of engineered exosome, we can achieve the dramatic increase of the brain fluorescence signal. Because the exosome is labeled with the fluorescence, this fluorescence represents the exosomes that are reached in the brain parenchyma. So we didn't know exactly how much in exosomes in quantitative manner, but by using this qualitative method, we now know that we can almost increase the 30-fold increase of brain penetrance by introducing this BBB targeting sequence on the surface of exosome. So by using the radioisotope method, the PET imaging or gamma counting, we are going to analyze more quantitatively of our brain targeting exosome, how much percent of exosome can reach the brain parenchyma compared to just the naive exosome. As I mentioned, naive exosome can pass the BBB only 1.1% of total exosomes. So from now on, I'm going to introduce our lead pipeline, ILB202, the anti-inflammatory exosomes. So these are our current pipelines. As I mentioned, the first pipeline, ILB202, is targeting acute and chronic inflammatory disease. And we are also developing the oncology and the CNS targeting exosomes in the preclinical stage. The ILB202 is exosome loaded with so-called super repressor I-kappa B. The super repressor I-kappa B is one form of dominant active inhibitor of inhibitor kappa B protein. So this inhibitor kappa B protein usually grab the NF-kappa B, the key transcription factor for immune and inflammatory system. So even though NF-kappa B is a transcription factor, they stay in the cytosol in the normal state because the I-kappa B grab and block the nuclear entry. But in the presence of pro-inflammatory signal like TNF, interleukin-1, or other LPS, the I-kappa B is readily phosphorylated and then degraded by ubiquitin proteasome pathway very rapidly, and then release the NF-kappa B, and they can enter the nucleus and do their job. But our dominant active form of I-kappa B cannot be phosphorylated because we changed the serine residue to alanine. So because of that, even in the presence of pro-inflammatory signal, they can still grab the NF-kappa B, can act as a very strong inhibitor of the immune system. So we are able to make the engineered exosome loaded with our protein of interest, and then test in vitro efficacy first. And we compare our ILB202 with just a recombinant super repressor I-kappa B as a negative control. Even though they can act as a strong inhibitor, this recombinant protein cannot work because they cannot pass the cell membrane easily. So even though we treated them with very high doses compared to our exosome, we don't see any inhibitory activity. But we can see the dose-dependent inhibition of, in this case, the LPS-induced TNF released in THP1 cells by our exosome. And as I mentioned, these are NF-kappa B, the immunostaining. In the normal state, you see mainly the P65, the major subunit of NF-kappa B, is in the cytosol, not in the nucleus. But after TNF stimulation, within 15 minutes, you can see the nuclear translocation. But by pre-incubation with our ILB202, you can see the nuclear translocation is almost blocked. And the DNA binding activity in nuclear extract of these cells is also blocked. So these collectively indicate our ILB202 is very active in vitro. And how about in vivo? So we have collaborated with many different groups to show our in vivo efficacy of our ILB202 in different animal models. The first one has been published last year in a septic animal model. The sepsis has been induced by just injecting the lethal doses of LPS or the CLP surgery, the circumligation and puncture, to induce the peritonitis and subsequent sepsis. But with all these different models, we can see the dramatic protective effect of our ILB202 in this animal model. And also, as a PD biomarker, the pro-inflammatory cytokines in the plasma can be reduced by our ILB202. And also, because we have seen very strong organ protection, mainly the kidney, in the sepsis model, as a follow-up study, we have focused on ischemic reperfusion kidney injury and the role of ILB202 in this animal model. And the clinical setting for the acute kidney injury is mimicked in the cardiac surgery. So we included the prevention study because most of the cardiac surgery is elective surgery. And very surprisingly, the prevention and intervention study, we can see almost the same protective effect. And as a mode of action study, we have tested how the exosomes can reach the kidney, because we know that in the normal state, the kidney is not a major target. The exosomes, once administered systemically, about less than 2% go to the kidney. But in case of acute kidney injury, more exosomes, we don't have the quantitative data, but qualitatively, more exosomes can reach the exosome, and most of the exosomes are uptaken by the immune cells infiltrated in the damaged kidney. And maybe that's why we see the protective effect in acute kidney injury model. And lastly, in collaboration with Texas group, who has been working the role of the exosome in the pregnancy, because we have seen the exosome can pass the placenta barrier, so we next tested whether our anti-inflammatory exosome can work in the pregnancy model. So we have tested the role in the inflammation-induced preterm birth model. So here, the preterm birth can be induced in rodent model by injecting sublethal doses of LPS in the pregnant mice. And here, at the day 15, they inject LPS, and you can see within 24 hours, they gave birth to all the pup as in a dead form. This is a preterm birth. But as you can see here, by injecting our ILB202 in maternal side, you can protect them in some way. And mean average delay is about 24 hours in mice system. That correspond to almost 2 or 3 weeks in human. And more important thing is, when we inject 1/10 of the doses of LPS here, the mice didn't give the preterm birth. They can give full-term birth. But as you can see here, the low dose LPS injected mice gave birth very low birth rate is intrauterine growth retardation. But this fetal problem can also be successfully blocked by our anti-inflammatory exosome. Okay, this is almost the last slide. So I want to introduce our ILIAS Biologics. So I have founded the company about 6 years ago. So right now more than 70 employees are dedicated to develop the first in human exosome-based therapeutics. So in the therapeutic areas such as inflammatory disease, cancers, and neurodegenerative disease. And our platform technology, as I have introduced, we can load the protein drug into the exosome, and we can actively targeting our engineered exosome to the specific type of cells. And I didn't have enough time, but we are able to produce the GMP grade exosome in large quantity for clinical study. So these are our major business partner program. So we are happy to work with other collaborators to study new novel therapeutic cargo that can be loaded into the exosome or other therapeutic area. Or we are very happy to expand our already developed ILB202 or other pipeline in different areas of indication. And I'm happy to answer the questions from the audience.